Namaskar and good morning. Welcome to CEC Gurukul lectures. I am Dr. Kumar Shantanu from Department of Botany, Deshbandhu College, University of Delhi. Today we are going to get introduced to a very unique group of plants called as Bryophytes. Bryophytes are unique in terms of their origin, in terms of them being the pioneer in any ecosystem, and also in terms of the diversity of morphology they exhibit. Bryophytes are most abundant in moist, temperate and tropical areas along the edges of streams and wetlands. Some of them even inhabit deserts or on dry exposed rocks. Mosses are dominant vegetation on rocky slopes also, above the timberline in mountains. Bryophytes, they can withstand severe cold of Antarctic continents. A few of them are aquatic or even grow on rocks that are splashed by oceanic waves. However, none of them are truly marine. Bryophytes in general represent the most primitive land plants. They require and are dependent on external water as medium for transfer of male gametes. They also exhibit a sharply defined heteromorphic alternation of generations. The gametophyte is dominant and nutritionally independent phase, whereas the sporophyte is nutritionally dependent on gametophytes. Asexual sporophytic phase is associated with the sexual phase of gametophyte. Majority views suggest that bryophytes arose from algae and probably from some green algae. Molecular systematics and study of cell structure tells us that more than 1 billion years ago, a heterophytic protist acquired an endosymbiont, and a photosynthetic descendant of such ancient protist evolved into red alga and green algae. The first event of endosymbiotic process took place between a heterophytic eukaryote and an autotrophic prokaryote. This event led to two lineages. One became green alga and second moved towards becoming red alga. The second event of endosymbiosis took place between a green alga and another autotrophic autotrophic prokaryote. Now this secondary event of endosymbiosis gave rise to two different lineages. One gave rise to euglenids and the other gave rise to chlorarachnophytes. It is chlorarachnophytes which are considered to be the ancestors of modern day green alga. This event took place at least 475 million years ago, and the lineage that produced chlorarachnophytes, which is a type of a green alga, gave rise to land plants. Researchers have identified the descendants was charophytes as the closest relative of land plants. Now let us understand some of the general characteristics of bryophytes. The division Bryophyta includes the simplest and the most primitive member of land plants that lack roots and do not have a vascular system. There are some mosses that have a primitive system of tubes that conduct water and food. The water conducting tubes are called hydroids. They have elongated, thick, dead cells and contain polyphenolic compounds, but they are not lignified like tracheids and vessels that are characteristic of vascular plants. The food conducting tubes are called leptoids and they are connected through plasmodesmata. Here in this diagram you can see the hydroids and leptoids shown with their radial sections. A single plant is very small, hardly a few centimeters in size. It seldom grows into a large plant because of lack of supporting tissue or mechanical tissues. Thousands of such tiny moss plants often grow together and give a thick green carpet-like appearance. As far as the reproduction in bryophytes are concerned, there are two prominent modes of reproduction: asexual reproduction and sexual reproduction. When we talk about asexual reproduction, it can take place in a bryophyte by three ways. The first way of asexual reproduction in bryophyte is by growth and branching, followed by death and decay of older parts. As you can see in this diagram, a bryophyte, usually a thalloid body, they grow with their apical portions and they dichotomously branch. As they grow and branch further, the posterior end starts becoming dead and they decay. As this decay keeps on rising, this decay reaches to the branching point. Once the decay reaches to the branching point, the original parent thallus gets divided into two different fragments, and each of these fragments can further give rise to a new thallus by the help of apical meristem and dichotomous branching. And this process keeps on getting repeated, and thus the thallus divides from 1 to 2 and from 2 to 4. The other method of asexual reproduction is by the separation of whole organs and the regeneration of new plants from them. There are certain bryophytes whose thalloid body give rise to structures such as tubers. Tubers are undifferentiated meristematic mass of cells which are covered with certain drought resistant or water certain tissues which do not allow water to desiccate. Similarly, there are certain adventitious branches also developing. These tubers and adventitious branches, they get all the more separated from the parent thallus and they germinate and give rise to new plant body. The third means of asexual reproduction is by the means of specialized units of propagation, also termed as gemma. Gemma are vegetative structures confined in a structure called as gemma cup. A gemma cup contains multiple gemmae. Each of these gemma, they germinate and give rise to two thallus in opposite direction. So from each gemma, two thalli germinate and thus the parent plant propagate through asexual reproduction. The first mode of asexual reproduction is very widespread in most thalloid liverworts. However, the separation of whole organ is the most common and effective means of spreading a plant among mosses. The gemmae is a fairly common mode of asexual mode of reproduction in some of the liverworts. Bryophytes show two distinct and well-defined phases of life cycle: sexual and asexual. Both of these life cycle phases follow each other. The gametophyte is haploid and produces gametes, whereas sporophyte is diploid and produces spores, which upon germination give rise to a new gametophyte. Now let us talk about sexual reproduction in bryophytes. Sexual reproduction in bryophytes is always oogamous. Although the bryophytes are land plants, their dependence on water is mandatory, and to complete the sexual cycle they require water. This feature of sexual reproduction in bryophytes is considered as one of the reminiscent of their aquatic ancestry. Because of this feature, bryophytes are often termed as amphibians of plant world. The biflagellate motile male gametes, the anthrozoids, travel to the non-motile female gamete, the egg, enclosed in an archegonium through the medium of water. This medium of water can be source coming from water stream, it can be a raindrop, or it could be the moisture which is aggregated from the surrounding by any moss plant. The diploid zygote, the product of fertilization, is formed within an archegonium. Now this diploid zygote is always in contact with the gametophyte. As you can see in this diagram, the gametophyte bears an archegonium at its apical position. Upon germination, upon fertilization, the egg cell gives rise to zygote, which after mitotic division gives rise to elongated embryo. This elongated embryo will further divide mitotically and give rise to a complete sporophyte. Even though the sporophyte is completely mature, it is always in physical contact with the lower gametophyte. This sporophyte is nourished by maternal tissue, that is the tissues of gametophyte. This phenomenon is termed as metrotrophy, that is food derived from mother. The sporophyte is diploid and produces haploid spores. The haploid generation alternates with diploid generation. Both the gametophyte and sporophyte may be several centimeters in length, but gametophyte is the dominant generation. The gametophyte may be thalloid, leafy thalloid, or has an axis differentiated into stem-like and leaf-like structures which lack xylem and phloem. In this diagram, we can see the example of Rixia, Anthoceros, or a moss. In all of these different types of bryophytes, the dominant phase is gametophytic phase, and none of them has a well-defined leaf or well-defined stem, and all of them lack any proper conducting strand such as xylem and phloem. These leaf-like structures, even when they are present, are part of gametophyte, whereas in vascular plants the leaves strictly develop on sporophyte. The adaxial surface of some thallus in liverworts, for example in Markensia, are specially modified to reduce water loss to the atmosphere. This adaxial surface also possesses specialized pores to increase carbon dioxide permeability. Here you can see a diagram of Markensia surface, and the surface has certain pores. These pores facilitate exchange of carbon dioxide. They also reduce the desiccation of thallus by not allowing water to evaporate from all the surface area. The gametophyte of a moss is green, photosynthetic and nutritionally independent, and they anchor to the soil by unicellular or multicellular filaments called rhizoids. Rhizoids appear like roots, but unlike roots, they lack vascular tissues and are much simpler in structure. Rhizoids mainly help as anchor. The intake of minerals and water is mostly and directly from the surface of the gametophyte. Here in this diagram, you can see the Fiscometrilla gametophyte, which has multiple number of rhizoids. These are multicellular branch structures, but they lack any xylem or phloem. Sexual reproduction in bryophytes. Let us understand the basic process of sexual reproduction in bryophytes. During migration from water to land, the need of protecting the gametes arose. So, possibly the sex organs developed a layer of sterile cells forming a jacket around gametes. The male and female reproductive organs are known as antheridia and archegonia respectively. The development of layer of sterile cells around the gametes, that is jacket, and the protective layer around the gametes was one of the land adaptations when bryophytes evolved from their aquatic ancestors. An antheridium in bryophyte consists of a single layer of protective sterile cells enclosing the mass of enterozoid mother cells or androcytes, each of which gives rise to a single biflagellated motile enterozoid. The position and shape of antheridia varies in different species. In this diagram, which you can see on the you will be seeing on the screen, there is an antheridia which is embedded inside the gametophyte thallus. The antheridia is surrounded by a single layer of sterile jacket. Inside the jacket layer, there are multiple fertile cells. These multiple fertile cells are also called as enterozoid mother cells or androcytes. These enterozoid mother cells, after metamorphosis, give rise to biflagellated spermatozoids or sperms. The archegonium is so characteristic of bryophytes, pteridophytes and gymnosperms that these three groups are collectively known as archegoniates. The archegonium is a multicellular, more or less flask-shaped structure. Its swollen base portion is known as venter and the upper elongated portion as the neck. It consists of an axial rows of cells surrounded by a sterile jacket. The axial row of cells can be distinguished into neck canal cells, a venter canal cell and a single large basal cell. This single large basal cell is termed as the egg or the oosphere. On the top of archegonial mouth, there are two swollen cells also termed as cover cells. These cover cells help guiding the spermatozoids chemotactically during the process of fertilization. The chemotactic attractant released by these cover cells allow the movement of enterozoids or the sperm cells, and these sperm cells move all the way down to the neck cells by dissolving the neck canal cells. There is one venter canal cell and thus finally they reach the egg cell. Upon reaching the egg cell, they fertilize the egg cell. The archegonium provides nourishment and protection to the egg and after fertilization to the developing embryo. The nutrient transfer from gametophyte and sporophyte takes place across cell walls through extensive labyrinth of highly branched cell wall. In this diagram, you can see two cells lying adjacent to each other. This cell is represented by GTC, which means gametophytic transfer cell. This gametophytic transfer cell has large vacuoles. At the same time, the cell wall is very much folded. This labyrinth structure increases the surface area of transfer. This gametophytic transfer cell lies adjacent to the sporophytic transfer cell or foot transfer cell, abbreviated here as FTC. Now, in between FTC and GTC, that is sporophytic transfer cell and gametophytic transfer cell, lies placental space. This intercellular space between gametophytic transfer cell and sporophytic transfer cell is termed as placental space. So, across these two cell wall, there is a rapid and more transfer of nutrients, and thus the sporophytic tissue is nutritionally supported by gametophytic tissues. This increases the surface area of the plasma lemma across which nutrients are transferred, and the tissue can be considered analogous to the placenta of animal kingdom. Now let us understand the process of fertilization in brief in bryophytes. In bryophytes, since the male gametes are ciliated, they require water to swim in order to reach up to the neck of an archegonium and also for the passage through the neck canal to the venter. A single antherozoid fertilizes the egg and the zygote is formed. The zygote begins to grow at once and divide by mitosis. Further, repeated mitotic divisions lead to development of multicellular embryo. In this series of diagrams which you see here, you can see the gradual development of an archegonia. Upon fertilization, the seta elongates and gradually the capsule expands and finally the capsule matures. This process is a gradual, this series of diagram represents a gradual sporophytic development. Across this developmental process, when the capsule starts to grow, the stomata also develops and the maturation of spores start to begin. The embryo is not liberated initially and it is retained within the archegonia, which you can see here in this diagram. After fertilization, the basal portion of the wall of archegonium enlarges and becomes multilayered and forms a protective envelope around the developing embryo, and this protective structure which is formed after fertilization, the basal portion of the wall of archegonium, which enlarges and becomes multilayered and forms a protective envelope around the developing embryo, becomes calyptra. The embryo eventually grows into a sporophyte. This protective envelope, which is formed by the tissues of gametophyte, that is the cells of archegonia, they are known as calyptra. The development of sporophyte is very limited and the embryogeny is soon followed by spore formation. There is a very little gap in the time, in terms of time, between the process of spore formation and embryogeny. In fact, the process of spore formation begins as the capsule keeps on growing. So both the development of sporophyte as well as spore formation keeps simultaneously alongside. Unlike other land plants, bryophytes are not differentiated into stem, leaf and roots. Generally, the sporophyte is distinguishable into a foot, seta, and a terminal spore-producing capsule or sporangium. In certain species, the seta is absent and more rarely also the foot. The spore mother cells develop inside the capsule and they represent the last stage of sporophytic generation. Here in this diagram, you can see a sporophyte of Rixia which lack any foot or seta. It is made up of a single jacket layer called as amphithecium, and below this amphithecium there is a layer of nurse cell. Confined with this jacket and nurse cell, spore mother cells are found. These spore mother cells, they divide and give rise to a spore tetrad. This division is meiotic. So spore mother cells are the last representative of sporophytic generation in any life cycle of the bryophyte. The spore mother cells divide by meiosis to form tetrads of haploid spores, which usually separate from before their discharge from the capsule. In bryophytes, the sporophyte has no connection with the soil and it is wholly dependent on the gametophyte for its nutrients. In some of the bryophytes, the sporophyte may possess chloroplasts and are partly autotrophic. Now, let us start observing the classification of bryophytes. When we talk about classification of bryophytes, you have already learnt that all the eukaryotic, multicellular, autotrophic and basically terrestrial organisms were placed in the kingdom Plantae by R.H. Whittaker way back in 1969. However, bryophytes are morphologically simple, evolution-wise significant and ecologically important plants. They have posed a very challenging task before the taxonomists. A fairly acceptable classification which will be at once reasonably natural and clearly workable has not been easy to meet. Robert Brown way back in 1864 used the term Bryophyta for the group that comprised algae, fungi, lichens and mosses. Later, algae, fungi and lichens were separated from this group and collectively comprised Thallophyta. An independent rank, Bryophyta, was first proposed by Schimper in 1879 to include liverworts and mosses. Later, Eichler in 1883 recognized two subgroups, the classes Hepaticae and Massae in the bryophytes. Engler in 1892 subdivided each of these classes into three orders. These three orders were: the class Hepaticae was subdivided into Markensials, Jungermanials, Enthocyrotales, whereas class Massae was subdivided into Bryals, Entrials and Sphegnals. This was a basic outline of classification of bryophytes. Till this time, we have understood various basic features of the group Bryophyta, their various modes of reproduction and a very brief classification of bryophytes. I hope you have enjoyed this lecture. Thank you so much.